RNA generated from the viral genome is going to be translated to make the glycoproteins. It's going to be translated to make the glycoproteins that are going to coat the surface of the virus. Also, the capsid proteins are going to be made. The glycoproteins that are generated from the viral messenger RNA are actually going to be generated in the host endoplasmic reticulum. That endoplasmic reticulum with the viral glycoproteins as transmembrane proteins is transported through the Golgi out to the membrane of the infected cell. So the infected cell is coated with viral glycoproteins. The capsid proteins are going to engulf the RNA genomes that are being generated. And these RNA genomes inside the capsid is going to be carried out through the microtubules. You know those microtubule tracks I talked about a long time ago? These are carried out on microtubules to the plasma membrane, where they will be budded off because the plasma membrane is studded with viral glycoproteins and a whole new virus is made. Isn't that cool? This is what happens when you have the flu. So it doesn't explode your epithelial cells. Your epithelial cells just get taken over. They just get taken over with viral proteins, viral genomes, and viruses budding off of the surface of the cell. Okay? What protects you? What's going to protect you from this influenza virus? What's going to happen? Why don't you die? Your immune system. Your immune system is the only thing to protect you from viral infection. Antibiotics are only good against bacteria. Antibiotics are the only thing you have against bacteria. But viruses, you have to have a fully functional immune system to be able to battle against virally infected cells. So what's going to happen? As a result of all of these viral proteins on the surface of these epithelial cells, what's going to happen is you have your immune cells. Your immune system is sending cells in, recognizing these abnormal proteins on the cell surface, and what's going to happen is your immune system is going to dispatch not only the new viruses that are being released, but it's also going to dispatch the cell that's infected with the virus itself. Okay? So it's your immune system that protects you. Even though the flu will get a good start, your immune system will come to the rescue. Okay? May take a day or two, but nonetheless, the reason you don't die is because of your immune system. Okay? All right. What's interesting is back in 1918, Philadelphia was the epicenter of an influenza outbreak. 13,000 people died in Philadelphia within the first 2 weeks of October. There were so many people who died that there were not enough funeral homes, not enough graveyards to absorb them all. So there were more than 10,000 coffins piled around City Hall, down Broad Street here, in October of 1918. That would give you a creepy feeling, right? If that happened now. But it did happen. And I'm going to talk about this in our next lecture, that it was an interesting form of influenza that was infecting the population at that time. And these people did die. Death usually occurred about 3 days after symptoms, and they died as a result of lung hemorrhage. It was a very different form of influenza. It killed 13,000 people within the first 2 weeks of October of 1918, but it killed millions upon millions across the world. Okay, This was a serious epidemic. They're looking at a field hospital of individuals infected with the virus here. All right. And what happened in this case is what I've got shown here. All of your cells, and this is not in your book at all. A lot of what I'm telling you is not in your book. All of the cells in your body have what is called the major histocompatibility complex, class 1, on the cell surface. All of the cells in your body have on their surface major histocompatibility complex proteins, class 1. And what do these do? Actually, what these do is you have your proteins, normal proteins in your cells, actually do get chopped up into small peptides. These small peptides are carried over to the endoplasmic reticulum, and the MHC class 1 proteins displayed on your cell surface, they're transmembrane proteins. So they're actually translated, or they're actually fully translated into the endoplasmic reticulum, and these peptides that are chopped up, normal peptides chopped up in your cells, are incorporated with the MHC class 1 transmembrane proteins in the endoplasmic reticulum, and they're transported to the cell surface. This is what your immune system uses to recognize your cells. This is these little normal peptides being displayed on your cell surface by the MHC class 1. This is also called HLA antigen. It's also called the HLA antigen. This is how your immune system recognizes your own cells. And if your cells are infected, if your cells are infected with viruses, whether it's an influenza virus or some other type of virus, what's interesting is viral proteins get chopped up, they get incorporated, and they're being shown on the cell surface. And these viral peptides of an infected cell, if they're on the cell surface, they're going to be targeted by the immune system. And that's the reason why so many people died of the influenza epidemic back in 1918, is because in these cases, the immune response to the infected epithelial cells in the lungs was so vigorous that the immune system itself was responsible for the death of the patient. Okay? What I also want to bring up to you is the concept of what some of these proteins are that are on the virus themselves, and that's hemagglutinin and neuraminidase. Hemagglutinin and neuraminidase are actually on the surface of the virus, and that's the H1 and N1 that you know about. You hear about the different influenza strains that infect individuals. That is the reflection of what type of hemagglutinin and what type of neuraminidase is on the surface of the virus. So this year, this past flu season, everybody was getting injections for hemagglutinin H1 and N1. And it turned out the primary flu virus this past flu season was an H1N5. And so therefore, it didn't help to be inoculated against H1N1 when the primary flu virus this flu season was H1N5. So there are various different types of hemagglutinin, there are various different types of neuraminidase, and depending on the hemagglutinin or the neuraminidase, that's the strain of virus that we have to look for every flu season. Okay? Do we understand this concept? Can I explain anything again? Not in your book. Okay? All right. Okay. Well, it never gets any better. Now we're going to talk about human immunodeficiency virus. In the case of human immunodeficiency virus, what you're looking at is a retrovirus. The human immunodeficiency virus is an envelope virus. It has a phospholipid bilayer with glycoproteins. Within the viral particle, you have a capsid or protein envelope in here. And inside that, you have the RNA genome. It's a single-stranded RNA genome. And associated with the RNA genome is an enzyme, a viral enzyme called reverse transcriptase. Okay, so the human immunodeficiency virus, what does it do? It actually recognizes helper T cells. Helper T cells are actually the immune system cells that are your first line of defense against any kind of invading bacteria or virus or foreign protein. Helper T cells will be exposed to bacteria or virus, and what they do is they activate proliferation of what are called cytotoxic T cells and also B cells. These are the cells. The B cells generate antibodies that are shed into the blood. Cytotoxic T cells, these are the T cells that actually will attack the infected cells displaying abnormal peptides. The cytotoxic T cells are what are responsible for that. Okay? So HIV, human immunodeficiency virus, what does it target? It targets the helper T cell. And the helper T cell is the first immune cell that is going to establish an immune response against bacteria, viruses, or foreign proteins in your system. So by targeting the first line of defense in our immune system, that's how it's called the human immunodeficiency virus, because what it does is it cripples the immune system of the infected individual. Does that make sense to you? Okay. All right. So the HIV target is the helper T cell. What does it do to this helper T cell? Well, it recognizes certain proteins on the helper T cell that allow for these glycoproteins on the envelope of the virus to bind. And then it's an envelope virus, meaning that once the glycoproteins bind to the target cell, then the viral phospholipid bilayer will fuse with the phospholipid bilayer of the target cell. The viral capsid with the RNA genome and the reverse transcriptase goes into the host cell cytoplasm. The capsid is removed, and then you have the viral RNA associated with this reverse transcriptase. The reverse transcriptase is what you call a RNA-dependent DNA polymerase. It's an RNA-dependent DNA polymerase. It is associated with the viral RNA genome, and what it will do is it will make a DNA molecule using nucleotides. Using nucleotides present in the host cell, it will make a DNA molecule. Then what will happen is the RNA from this RNA-DNA hybrid that's generated, this RNA will be degraded, and the transcriptase will make a second strand. So now you have a double-stranded DNA. This double-stranded DNA actually gets incorporated into the helper T cell chromosome as a provirus. As a provirus, what's going to happen is the host cell RNA polymerase, is going to transcribe the viral genome incorporated into the host cell chromosome, or the helper T cell chromosome, to make more chromosomal DNA, or to also make more chromosomal DNA, as well as more messenger RNA from the viral chromosome. So the viral DNA that's incorporated is going to be for the next genome. It's going to make messenger RNA to be translated into viral proteins, including the glycoproteins and the capsid proteins. These are going to be the viral surface proteins or glycoproteins are generated as the endoplasmic reticulum, shipped out to the cell surface. The capsid proteins will actually engulf the single-stranded genome as well as the reverse transcriptase. And new viral particles are actually budded off the infected cell, as you see here. And that's what we're looking at in this slide here. This is an electron micrograph of a human immunodeficiency virus entering a helper T cell, which is actually a white blood cell, but it's actually a helper T cell that the HIV will bind to and enter. And then what you're looking at here, after the cycle I've just gone through, then the virus will bud off as a new viral particle to now go and infect other cells in the body. Okay. Are we okay with viruses? Did I repeat anything? Like I said, it never gets good, it only gets worse. What's even worse, and I'm going to spend a lot more time on viruses next Monday, but what's even worse are prions. Prions are actually infectious proteins. They were identified as being the cause of a degenerative brain disease called Creutzfeldt-Jakob disease, or SCRABE. What happens is usually prions are usually eaten. It's usually in the form of some kind of meat that contains prion proteins. What prion proteins do is they get incorporated into the brain, and they will actually bind with normal proteins in the brain to cause aggregates that kill the brain cells. What's scary is prions cannot be inactivated through high cooking temperatures. There's at least a 10-year incubation for the development of the disease. There is no known treatment or cure. This work actually won the Nobel Prize back in the early 1980s, because it was the first time that the idea of an infectious particle that was not a virus or a living organism was identified.